Good afternoon, everybody. My name is Hans Herring, and today I'm going to be presenting my final project for my epidemiology course. And this is, of course, at West Coast University. The article which I'm going to be presenting about is one that was suggested by my professor, Helena Flores. So to begin, I'm going to be talking about TBI, which is traumatic brain injury. So, traumatic brain injury alters brain function and brain pathology. It is caused by traumatic injury to the head or body, such as in major and multiple impacts in football. The severity of damage from the injury is influenced by factors such as the force of impact and other attributes of the incident. Approximately 70,000 to 90,000 people each year get TBI, so severe that they are left with irreversible, debilitating loss of functioning. The present study being examined tests the neuroprotective efficacy of Velcade, a proteasome inhibitor, in a model of TBI in rats. Sensory motor and spatial memory tests were used in the present study to evaluate the rat's function response to injury and treatment after traumatic brain injury. Results from this study demonstrate that Velcade improves spatial learning functioning after TBI. Treatment with Velcade reduces cerebral inflammatory response, and treatment with Velcade promotes microvascular integrity. So one of the questions was, why did the scientists perform the study? Were there any specific or possible connection between the exposure and the disease? Well, in a traumatic brain injury, an alteration in brain function or other evidence of brain pathology caused by major or traumatic injury to the head or body, the severity of damage from the injury is influenced by factors such as the force of impact and other attributes of the incident. There are many types of incidents that can cause TBI. Some of these include falling, automobile collision, violence, sports injury, and explosions. It is important to note that there are many problems with TBI data collection. Therefore, data that is present in this essay and research studies may not accurately predict true incident rates. For example, patients with mild TBI may never go to a hospital for treatment because they may not believe it to be severe enough. Others who do end up going to the hospital may be discharged from the emergency room or the ER without adequate documentation, and those with severe TBI who pass away at the scene may not be fully accounted for in TBI epidemiologic studies. The prevalence, therefore, is not accurately accounted for. Furthermore, the definition of TBI, including its ancillary symptoms, vary from doctor to doctor and case to case. According proceedings from the National Institute of Health Consensus Development Conference on Rehabilitation of Persons with TBI, 1998, approximately 70,000 to 90,000 people each year get TBI so severe that they are left with irreversible, debilitating loss of functioning. TBI causes a strong inflammatory response that is partially responsible for acute pathological processes observed after TBI, so like abnormal mechanisms and forms involved in the dysfunction of tissue and organs, and again observed after TBI. Proteasome inhibitors are drugs that help reduce blood clots and inflammation, thus protecting vascular functioning. Proteasome inhibitors are being studied in the treatment of cancer in animals. So here's another question is, what was the hypothesis under investigation? The present study tests the neuroprotective efficacy of Velcade, a proteasome inhibitor, in a model of TBI in rats. What were major techniques used in the study? Well, in methods, the present study explores the efficacy of Velcade using a total of 32 adult male Wistar rats, each weighing about 300 to 500 grams each. The rats were split into two experimental groups, each containing 16 rats. The first groups consisted of rats that were sacrificed 35 days after traumatic brain injury was induced. Of this group, A received 0.2 milligrams per kilogram of Velcade to treat the TBI. The other A received 0.2 milligrams per kilogram of saline to treat the TBI. The second group consisted of rats that were sacrificed 24 hours after traumatic brain injury was induced. Of this group, 8 received 0.2 milligrams per kilogram of Velcade to treat the TBI. The other 8 received 0.2 milligrams per kilogram of saline to treat TBI. So now we're going to talk about the measures. Various tests were performed to evaluate the function response to injury and treatment after TBI. This section explains all the tests performed. A sensory motor functional test was performed using the Modified Neurological Severity Score, also known as MNSS. In this test, rats were given a combined score on the following test: motor, sensory, beam balance, and reflex. The spatial learning memory test was given to the rats was a modified of the Modified Morris Water Maze, also known as MWM, in which the rats were timed in finding their way to a hidden platform. Tissue preparation was performed to calculate the volume of the lesion in the rats. Immunohistochemistry was carried out to examine the inflammation on the brain, stability of cerebral microvessels, and the durability of neurons after TBI. Western blot analysis was performed to identify specific amino acid sequences in proteins. Lastly, an enzyme-linked immunosorbent assay, also known as the ELISA, was carried out on the same brain tissue from the Western blot to quantitatively analyze protein. So now we're going to talk a little bit about the test procedure. As previously mentioned, sensory motor and spatial memory tests were used in the present study to evaluate the rats' function response to injury and treatment after TBI. This section summarizes the test procedure. First, a sensory motor functional test was performed on all rats before injury and on the days 1, 4, 7, 14, 21, 28, and 35 after TBI. Then all rats were anesthetized intraperitoneally with chlorohydrate to induce injury with a CCI device. Next, the 24-hour rats were sacrificed. Then the spatial learning memory test was performed on the remaining rats on days 31 to 35 after TBI. Then, the 35-day rats were sacrificed. Next occurred tissue preparation to calculate the indirect lesion area in the lesion volume presented as a volume percentage of the lesion compared with the contralateral hemisphere. Immunohistochemistry was then carried out to examine the cerebroinflammatory response after TBI. Then a Western blot analysis was performed to detect specific proteins in tissue samples. Lastly, an ELISA following the data analysis. So another the question was, what were the major results, and do they support or negate the hypothesis, and do they point to further future studies? Well, for the results, there were a lot of results. Compared to the scores of the saline group, treatment with Velcade significantly improved sensory motor function on days 4, 7, 14, 21, 28, and 35. Spatial learning function was significantly improved in rats treated with Velcade compared with that of the saline treated groups on days 32, 33, 34, and 35. The lesion volume of the Velcade treated group was significantly reduced compared to that of saline treated group when examined on the 35th day. Treatment with Velcade significantly reduced the loss of neurons in the dengue gyrus in the hippocampal CA3 region compared with the saline group in rates 35 days after TBI. Treatment with Velcade 2 hours after TBI significantly reduced the number of intracellular adhesions molecule 1 immunoreactive vessels and MPO immunoreactive cells detected 24 hours after treatment compared with the saline treated group. Treatment with Velcade at 2 hours significantly increased collagen 4 immunoreactive vessels and reduced MMP9 immunoreactive vessels in the ipsilateral cortex compared with the saline treatment group examined at 24 hours after TBI. These data demonstrate that Velcade improves spatial learning functioning after TBI. Treatment with Velcade reduces cerebral inflammatory response and treatment with Velcade promotes microvascular integrity. In conclusion, it appears that Velcade preserves the neuronal structure and or function and may be valuable in treatment of TBI. There is no formal hypothesis in the present study in which result would support or negate, as there are no prior studies that explore the effect of Velcade on TBI. So now I'm going to be talking about some of the strength and limitations of this article. So here's for some of the strengths. There were actually several. First, researchers were able to induce traumatic TBI in rats and measure rats' function response following TBI, including statistical validity. Second, the study contained an immense level of detail with regards to the method and procedure. Third, the reference used, scholarly and peer-reviewed articles indicate this study is of high quality and significance in the field of epidemiology and medicine. Lastly, their article was extremely interesting and great at keeping the reader's attention. So now for some of the limitations. Although the present study was very interesting and well written, it was also contained some limitations. First, the Velcade was only tested on rats, yet the analyses are meant to illustrate potential human benefits. Second, the study was convoluted, encompassing a great deal of jargon, definitely written for an audience well versed in the structure and function of the brain, drugs, advanced statistical analysis, and medical terminology and procedures. Third, further tests need to be conducted to study the effects of Velcade on endothelial cell gene expression, which provokes vascular inflammatory responses, thrombosis, and blood-brain barrier, also known as BBB damage. Lastly, additional rats are needed to study the therapeutic window and dose responses for Velcade treatment of TBI. So the reason I chose this article to review was because for many reasons. First, because its association with football players. Because I played football in high school and I was really curious to know about what type of injuries they may receive. A couple of months back, I had watched a documentary on Netflix, A League of Denial: The NFL's Concussion Crisis by PBS. The documentary explained the correlation between traumatic football impacts and mental brain illness. I was shocked to learn about the prevalence in CTE diagnosis among football players. I am a big fan of football, and like I said, I played it, but after watching this documentary, I reevaluated my perception of the game and its effects on players. Former football players had developed Alzheimer's, depression, and dementia. Growing up in SoCal, Junior Shaw was one of my favorite football players. In 2012, Junior Shaw took his life because of a physical abuse and cerebral collision he endured while playing football. He was diagnosed with CTE along with 49 of 50 other former football players whose brain were also examined upon death. In 2013, the National Football League reached a settlement with 18,000 former players in the amount of $765 million. This documentary created a third knowledge for TBI. I really enjoyed reading this article about TBI and Velcade. Sometimes I had difficult comprehending the details, but was able to understand the overall procedure and meaning. Although there were several times where I had to look up terminology, it was very interesting. Clearly written and very detailed in the method, procedure, and drugs used. It was helpful to have these explicit reports or reporting. Thank you.